Hi, my name is Zara Purohit, and my project is managing diabetes with nanoparticles. My mentors were Nelson Hawkins, Dwana Jones, and Manuela Sushnita at the Methodist Research Institute. My research objective is to create a nanoparticle treatment for patients with diabetes. Before I talk about diabetes in context with nanoparticles, let me introduce what nanoparticles are. Nanoparticles are particles of matter that are between 1 and 100 nanometers in diameter. There's currently huge potential in utilizing nanoparticles as they're very small and able to enter the body and be biocompatible to medically improve one's health. Currently, there are many applications of nanoparticles, such as in electronics, fuel solar cells, batteries, and space. For my project, I'm going to be focusing on the applications of nanoparticles in medicine in areas such as drug delivery, diagnostics, antibacterial treatments, wound healing, imaging, and cell repair. There are three different types of nanoparticles: organic, which are carbon based and include polymeric based nanoparticles, liposomes, and carbon nanotubes. Inorganic, which are non carbon based, include gold, silver, iron oxide, and quantum dots. And finally, hybrids, which are a combination of these materials and include a mixture of organic and inorganic materials and nanogels. I've decided to go with an organic nanoparticle as they're typically more biocompatible due to their material. I've chosen Kydzen, which is a type of polymeric based nanoparticle. And some advantages of are that it is biocompatible, it is easily degradable, it erodes under acidic conditions, it can easily be fine tuned, it is a glucose dependent release, which means it'll release the drug depending on the levels of glucose in your blood. It is a sustained release, which means it won't release the drug altogether, it will do a little bit and a little bit over a period of time. So the method of fabrication of Kydzen is first to prepare Kydzen, then you would prepare and add the cross-linking agent, which in this case is tripolyphosphate or TPP. You would stir the solution so that it undergoes ionic gelation. And in this case, in this picture, you can see the positively charged Kydzen and the negatively charged TPP are cross-linked together to create an aggregate. You would separate the cross-linked nanoparticles from the ones that didn't cross-link, and you would add a coating to these nanoparticles. I've chosen polyethylene glycol or PEG because this coating creates a superior loading capacity of insulin, it enhances stability, it lowers blood glucose levels without causing hypoglycemia or low blood sugar, it improves intestinal absorption and release behavior of the hormone. So, what is diabetes? Diabetes is a condition in which the body does not properly process food for use as energy. Insulin, a hormone created in the pancreas, helps glucose get into the cells of the body. So, in patients with diabetes, a sufficient amount of insulin is not created. Therefore, sugars build up in their blood. Currently, diabetes is being treated with diet control, exercise, home blood glucose testing, and insulin injections. Type 1 and type 2 diabetes. So, the difference between these different types of diabetes is that in type 1 diabetes, there is no insulin created, and in type 2 diabetes, the cells are not able to respond to the insulin created. So, in both cases, the cells do not absorb the glucose. So the goal of my project is to create a hypothetical nanoparticle that contains metformin that allows blood sugar levels to decrease. Nanoparticles and diabetes. So with my nanoparticle treatment, we would be able to eliminate the use of insulin injections. You would not have to use this daily. It would be a superior loading capacity of insulin and is a glucose dependent release. After creating this nanoparticle, I would use these techniques to analyze the particle in vitro. With, with the cell culture technique, I would grow cells to test. And with these cells, using the NTS cell proliferation assay, It would allow me to, after a process, compare the absorbance readings with nanoparticle treated cells to untreated cells to see how many cells are still alive after the nanoparticle has interacted with them. Visualizing nanoparticle uptake allows you to see where the nanoparticle goes inside the cell. So, after creating this nanoparticle, it's important to test different attributes of the nanoparticle, such as size, surface charge, and concentration. The DLS, or dynamic light scattering, allows you to test the size of the nanoparticle. As you can see, smaller particles create a larger signal, and vice versa for larger particles. It's important to make sure you have the optimal size for your nanoparticle depending on what function it has. For my nanoparticle, I would like the size of the particle to be similar to blood cells or other cells in the body and be able to enter these cells with ease. This zeta potential analyzer allows you to find the surface charge of the nanoparticle by finding the difference of charge in a specific solution. Surface charge is important for the stability of nanoparticles in suspension and is also the major factor in the initial absorption of nanoparticles onto the cell membrane. Imaging allows you to visualize nanoparticles, so TEM, which is displayed on the bottom, or transmission electron microscopy shows a 2D representation of the nanoparticle, and SEM, or scanning electron microscopy, shows a 3D representation. After in vitro testing, we would conduct in vivo testing. So we would test this nanoparticle in diabetic mice. Again, you would test for the size using the DLS, the surface charge using zeta potential, and concentration of the drug using nanosite. You would see the overall effects on the mice, how has their health been affected, and of course, their blood sugar levels. So some findings. If I were to conduct this experiment in real life, if I was a researcher, I would use these techniques I discussed to test my nanoparticle and see if it functions as desired. This requires research funding and lab materials. Some questions that I do not know, as I'm unable to test it, is if other treatment would be required. Are there any toxic effects? Does it affect function as desired? Does it affect other parts of the body? Should I change the concentration of the drug? Should I perhaps use another drug? And many more. After I complete my research, I would make sure my nanoparticle is reviewed and approved by the FDA. Then it will be sent to hospitals for feedback and patient testing. Overall, diabetes is a highly prevalent illness in America, and as such, there's a high demand for a different form of treatment for these patients. Nanoparticles will allow for a novel method towards the treatment of diabetes and create huge positive implications for these patients. Here are my citations, and thank you so much for listening.